I'm Anna Raymond, and today I'm going to talk about comparative embryology. Though controversial at first, Darwin's theory of evolution is now accepted by most scientists. Comparative embryology, or the study and comparison of different embryos and how they develop, was crucial in the making of this theory. Early scientists found it difficult to tell the early embryos of different species apart. In fact, Carl Ernst von Baer, an influential embryologist, once had 2 embryos preserved in jars. He had forgotten to label them and discovered that he couldn't tell them apart. Well, how does comparative embryology support the theory of evolution? First, different organisms share similar patterns of development. Mammals and fish embryos both start off with pharyngeal arches that become gills in fish and the jaws, ears, and skull of mammals. This shows that mammals and fish are probably related. Also, many vertebrates have similarly composed pentadactyl limbs. Different animals have evolved to use them for different things: swimming, walking, and flying. If all 4-legged vertebrates have pentadactyl limbs, they must share a common ancestor. Triploblastic animals, or all multicellular organisms, except for a few vertebrates, all begin development with 3 layers: the ectoderm, mesoderm, and endoderm. We can take away from this that all triploblastic animals share a common ancestor. The second reason comparative embryology supports the theory of evolution is the presence of vestigial organs, which are body parts that either have no function or are not used for what they were originally used for. Human embryos have a tail at one point in development, but they go through apoptosis, or automated cell death. The tail has no use for us, so we must have it as a vestige from our ancestors. The now extinct dodo bird had hollow bones like animals that can fly. Therefore, even once structures are no longer needed, it takes many generations of evolution for them to go away. The third reason comparative embryology supports evolution is the fact that many organisms have similar homeotic genes. They are often called master genes and are in charge of the body's development. One class of homeotic genes is Hox genes. The order of these genes on the chromosome determines where body parts will be located on the organism. Mutations can cause body parts to be in the wrong place, like a fly having legs on its head instead of antennae. The order of the Hox genes is extremely similar between different organisms. In many vertebrates, there are 4 similar gene clusters for 4 similar parts of the body: Hox A, Hox B, Hox C, and Hox D. This suggests that the order generally remains constant while the animals evolve, pointing to a common ancestor. Our current knowledge wouldn't be the same without the field of comparative embryology. Not only do different organisms share similar structures and patterns of development, we can observe vestigial organs in embryos to learn about common ancestors. Also, the similarities in Hox genes can tell us which animals are related. All in all, it isn't surprising that von Baer couldn't find out which animal was on that little jar.